Hi, welcome back. Today we're covering Chapter 6, Acellular Pathogens. In this first section, we're specifically covering viruses. Chapter 6 is divided into 4 sections. In the first 3 sections, we'll talk about the viruses, their life cycles, and how to isolate, culture, and identify them. In the last section, we're going to cover material I briefly talked about in Chapter 1, viroids, virusoids, and prions. These are also acellular pathogens, although they're different from viruses. For this first section, viruses, we're going to look at a very general overview about viruses. By the end of this section, you'll be able to describe their basic viral characteristics, their genomes, a tiny bit about their life cycles. We'll focus more on that in the next section, and then how we classify viruses. Viruses were first discovered in the late 1800s, and it's pretty interesting because what was happening at the time were that tobacco plants were getting sick, and they called this tobacco mosaic disease. They took sap and extract from these sick plants and filtered that through filters that normally trapped bacteria. Then they took the filtrate, the liquid that passed through the filters, and exposed that to healthy tobacco plants. And it turned out that those plants would get sick subsequently after exposure. Dmitry Ivanovsky thought that maybe it was an extremely small bacterium making these plants sick because it still passed through those filters. It wasn't until later on, through additional study, that Phage, a Dutch microbiologist and botanist, eventually called it a virus. And this is actually Latin for poison. At first, they thought it was some kind of poison making them sick. They didn't know that viruses existed in the form that we understand today back then. It wasn't until the invention of the electron microscope that people were able to see viruses. Viruses are not cells. They're infectious acellular particles. A meaning not, so not cellular. They cannot reproduce on their own, and they actually have to hijack a host cell, which is why we call them obligate intracellular parasites. They have either DNA or RNA genomes, but never both within the same virus. Viruses are packaged into particles called virions, and this consists of genetic material, that DNA or RNA, plus a protective protein shell called a capsid. Some also have a lipid envelope that they steal from the host cell. This is a nice table from our book summarizing those characteristics of viruses. I wanted to point out that that membrane that some viruses have, that envelope, I mentioned comes from the host cell. It comes from the host cell membrane as the viruses bud out or exit the cell. And they are covered with viral glycoproteins to allow for attachment to the next host cell. And I wanted to compare these viruses to some obligate intracellular bacteria that we talked about in a previous chapter, because both of these can be obligate intracellular parasites. So why are bacteria considered cells and alive, whereas viruses are not? So bacteria are cells. They're prokaryotic cells because they still have DNA and basic structures like ribosomes and cell membranes, whereas viruses do not. They're acellular. For bacteria, they do have limited metabolism. So for these obligate intracellular bacteria, they do rely on a host for things like making ATP, but they can perform some basic metabolic processes themselves, whereas viruses cannot at all. They completely depend on their host. Binary fission can occur inside host cells for bacteria, but here again, viruses are relying on the host cell machinery, the host cell enzymes. Here, our genetic material is DNA for bacteria. Viruses have sometimes strange types of genetic material. They can have double-stranded RNA or even single-stranded DNA, which you don't see in other organisms. And here, of course, bacteria can respond to antibiotics. And here, viruses do not. So you can't take antibiotics for a viral infection. You need some kind of antiviral medication or hopefully preventative vaccines. And then how do these survive outside of the body, outside of a cell? Bacteria like chlamydia can form elementary bodies that are kind of similar to the endospores for some bacteria we talked about. They are dormant and allow for short-term survival outside of the host. Viruses are similar. They have that protein shell, the capsid shell, that allows them to survive until they infect a host. So the infectious agent of tobacco mosaic disease was able to pass through filters that normally trapped bacteria, and that's why they thought at first that it was a toxin. It wasn't a visible bacterium. Scientists thought it could be like a part or a toxin or a chemical poison released by something like bacteria, rather than what we know today is now called tobacco mosaic virus. Viruses infect all types of organisms: plants, animals, fungi, protists, bacteria, and even archaea. But most are host-specific and cell-specific. For example, bacteriophages, we use phage for short, only infect bacterial cells. Another example could be HIV in terms of cell-specific. HIV primarily attaches only to certain cells in our blood, like T cells. There are even respiratory viruses that can cause cold-like symptoms in dogs, but don't affect us, humans, such as the canine flu virus. So if your dog has that virus and then they lick your face, you won't get sick because that virus is specific for dogs. Viruses can spread in several different ways. One is through direct contact between people. Viruses can also spread through indirect contact through objects, also known as fomites. Fomites could be things like toothbrushes. If you share toothbrushes and one person is sick, the other person can get sick that way. Or maybe stuffed animals. If one child is sick and is touching and sneezing on their stuffed animal, if the next child picks it up and plays with it, they could get sick. We can also get sick through vectors, which are animals that can pass disease from one host to the next. And we have two types here: mechanical vectors and biological vectors. Mechanical vectors pass parasites from host to host through their surface. So a cockroach, for example, can be a mechanical vector if it's walking over trash and picking up different types of bacteria in the trash, and then they walk over your food, right? They can drop those bacteria onto your food, making you sick later on. Whereas biological vectors are themselves also hosts for the parasite. One example is, here is a mosquito. They're drinking blood from this person. Maybe this person has malaria and they pick up Plasmodium, the protozoan that causes malaria. And then they fly over to the next host and inject that Plasmodium into a healthy person, thereby making them sick, right? Transmitting the protozoan to that second person. Some viruses can even cross species, and when this happens, we get something known as zoonoses. So this can happen such as in rabies, when we get bitten or scratched by a bat, for instance, or through bird flu. We can also transmit diseases from human to animal, and then when that happens, that's known as reverse zoonoses. You've probably heard of antibiotic resistance, and these antibiotic-resistant bacteria, or superbugs, actually cause over 3 million infections and nearly 50,000 deaths every year in the US. Our book actually has lower numbers, but they're kind of outdated, so this is closer to what we currently have annually. One possible solution is phage therapy. I just mentioned that phage are viruses that target specific bacteria. And this actually is not new. It was first used way back in the 1900s, early 1900s. So in 1915-ish. But then it was abandoned. This practice was abandoned when penicillin came along. Now it's making a comeback because we have so many antibiotic resistant strains. So there are a few advantages to phage therapy. Phage are very specific. They only kill a specific type of bacterium. They usually will not affect our normal flora, the rest of our normal bacteria that are not making us sick. They're also less expensive to develop than new antibiotics. Phage are also used in food safety. So our book gives us an example of a a phage spray in 2006 that was used in deli meats to prevent listeria. So Listeria monocytogenes is a bacterium that you might see on things like cheeses, soft cheeses, and depending on the type of regulation, locally or nationally, foods that have been treated with phage might be labeled as bacteriophage preparation that's involved, or you might see it treated with antimicrobial solution, but it doesn't have to be labeled like this anymore. So I think the book says must be labeled, but I would say it's more like may be labeled depending on how the food was processed. So for this checkpoint, we don't have to be concerned about the presence of bacteriophages in our food because bacteriophages only infect bacteria. They don't infect human cells, and they're harmless to people and sometimes even used to prevent foodborne illness, like in that spray. In the second one, what are 3 ways viruses can be transmitted? We said direct contact between person to person, indirect contact between an object or fomite to the next person, or vectors. We have these insects or arthropods that can be mechanical vectors where they carry pathogens on their body surface, or biological vectors where they're carrying pathogens inside their body. And these are transmitted, for the biological vectors, they transmit the disease through bites. Viruses are important because they cause many diseases like flu, HIV, herpes, Ebola. Some are even linked to cancers, like human papillomavirus causing cervical cancer. Unfortunately, we still don't have as many effective drugs to treat viral infections. For instance, unlike bacterial sexually transmitted infections like chlamydia, which antibiotics might be able to cure, viral sexually transmitted infections like herpes cannot be eliminated. You just have to manage them with antivirals or vaccines. On the positive side, viruses can also be useful, such as in phage therapy. Viruses are tiny, on the order of nanometers, ranging from about 20 to 900 nanometers, much smaller than cells. Each variant has a nucleic acid core and a protein capsid. There are 2 general types. Naked viruses, also called non-enveloped viruses, have only a capsid outside of their genome, whereas enveloped viruses have an additional lipid membrane outside of the capsid, and this membrane comes from the host cell membrane. Finally, we have spikes on some viruses to help them attach to and enter cells, like the flu's H and N spikes. These stand for hemagglutinin and neuraminidase, and I'm not going to ask you about that though, just FYI. Influenza viruses are often identified by their H and N spikes. For example, H1N1 influenza viruses were responsible for the pandemic in 2009, and then many more years before that in 1918. Here we can see both naked and enveloped viruses. For the naked virus or non-enveloped virus up here, we have our genome, then our protein capsid, and then the spikes are on the capsid. Here is an example of an actual virus under the electron micrograph. And down here we have an enveloped virus. The genome is in here, then we have our protein capsid shell, and then out here is the lipid envelope with spikes embedded within the envelope. And on the left, they are showing us, it looks like HIV budding out of a cell. It's hard to see, but all these tiny little dots everywhere, those are the enveloped virus. Viruses come in 3 basic shapes. We have helical or rod-shaped, such as tobacco mosaic virus. Ebola also has this shape. Polyhedral, which is a 3-dimensional multi-sided shape. It can be icosahedral or 20-sided, such as in polio or rhinovirus. And then we have complex structures. Usually this means like a combination structure, such as bacteriophage, which I'll show you in a second. Pox viruses are kind of brick-shaped and don't really fit any of the other simpler categories. And here's our bacteriophage. You can see it has a pretty complex shape. It has that polyhedral capsid, but also this rod-shaped sheath, as well as tail fibers for attachment. And you can see how small it is compared to the cell. This is a bacterium, so I'm guessing the bacterium is going to be this huge cell relative to the virus. Here's another example where you can see more of the bacterial cell and many bacteriophage infecting that single cell. And this is my favorite because of their shape, but I guess many others love it too because you can buy phage jewelry and even phage planters. All right, going back to looking at what viruses have spikes, remember that both naked and enveloped viruses can have protein spikes for attachment. They help the virus attach to and then enter the host cell, such as the influenza H and N spikes. And a quick reminder about the relative sizes of cells. We did the gum line gram stain, and we saw that our epithelial cells were about 50 to 60 micrometers in diameter. Of the many bacteria we've seen, many of them are only about 1 micrometer long or 1 micrometer wide. But viruses, we're looking at the order of nanometers, where 1 nanometer is 1,000 times smaller than 1 micrometer. Viruses are not in the 3 domains of life, so they're actually classified by the International Committee on Taxonomy of Viruses using their system. Here, family names will end in viridae and genera end in virus, such as shown here, herpes viridae family, herpes virus genus. And this link will take you to current taxonomy of viruses. Another system is the Baltimore system that groups viruses by their genome or their replication method. And for the genome categories, we have double-stranded or single-stranded DNA, double-stranded or single-stranded RNA, and we'll talk about positive and negative in the next section. But we can also have informal groups of viruses, such as categorizing them based on their general genome or their structures like enveloped. So herpes viruses are in this general group of double-stranded DNA enveloped viruses. Common human viruses can be divided into whether they contain DNA or RNA, and all of these names are family names, as I can tell from that word right there, or that part of the word viridae. Herpes viridae can cause, or viruses within this family can cause cold sores or genital herpes. For papilloma viridae, these viruses can cause warts or cervical cancer. For orthomyxoviridae, some of these viruses can cause influenza. Rhabdoviridae can cause rabies, and viruses within this family, retroviridae, include HIV, the virus that causes AIDS. Don't worry about these other ones besides the ones I just talked about in the previous slide. Later on in the next section, we'll talk about why it's important that viruses are categorized by their genome. This is going to impact their viral life cycle, and we'll see that in section 6.2. We talked about 5 types of viral genomes: double-stranded DNA, single-stranded DNA, double-stranded RNA, and then 2 types of single-stranded RNA, positive sense and negative sense. And we're going to talk about what that means in the next section. So the ICTV also links viral classification to the International Classification of Diseases, or ICD, which is maintained by the World Health Organization. ICD codes are alphanumeric and track not only viral infections, but all medical conditions. These codes are used by clinicians, labs, insurance companies, medical coders, and even epidemiologists in order to diagnose, order tests, process claims, and then track mortality or death statistics. And here, morbidity means disease. So morbidity means disease and mortality means death. So ICD code, ICD again stands for International Classification of Diseases, and 2 locations where you might find one include medical records or insurance billing systems for diagnosis and reimbursement, or death certificates or epidemiology reports in order to record the cause of death and track disease statistics. This chapter, like the previous chapters, also has a clinical example. In this one, we have a patient, David, that was bitten by a dog and they're worried about rabies. You'll see lots of clinical connections here where they talk about diagnosing a viral infection by looking for the antigen. Antigens would be something on the virus surface that we could detect. Looking at antibodies, antibodies would be in the patient, showing that the patient has been exposed to the antigen, the virus, or you could even look for the nucleic acid present in the virus. So look for the DNA if it's a DNA virus, or look for the RNA if it's an RNA virus. You can do that through RT-PCR, and we'll talk more about that later. Treatments can include vaccines, antiviral drugs or immunoglobulins, depending on the virus. And again, we'll talk more about this throughout the semester as well. The study of viruses, or virology, is still evolving. We've recently discovered giant viruses like Pandora virus and Pithovirus that are larger than even some of the bacteria that we have. So this is kind of challenging the idea that all viruses are pretty small on that nanometer scale. We'll talk about CRISPR later on as well. CRISPR systems are powerful gene editing systems discovered in bacteria and archaea. And then we'll also look soon at mRNA vaccine breakthroughs. So using something called positive sense single-stranded RNA technology, we were able to develop many COVID vaccines. In general, zoonotic spillover is still important and a major concern, as we saw with SARS coronavirus 2 that causes COVID-19. Genetic studies showed that this virus is closely related to coronaviruses found in bats. So we're not exactly sure what happened, but the virus likely jumped to humans, either directly or through an intermediate host. And then finally, we have phage therapy revival. With the rise of superbugs or antibiotic-resistant microbes, bacteriophages are again being tested to see if they can be alternatives to antibiotics. All right, let's recap 6.1. General characteristics. They are acellular infectious agents, not made of cells. Viruses have to have a host cell to reproduce. Their genome is either DNA or RNA, but not both at the same time or within the same virus. They're surrounded by a protein capsid. Sometimes they have a lipid envelope, and they lack their own metabolism, right? Viruses depend on a host for replication. For viral genomes, we talked about how they can be DNA or RNA within the capsid, but never have both at the same time. Those molecules can be single-stranded or double-stranded. And for RNA genomes, they can be positive sense or negative sense. For the third one, general characteristics of viral life cycles, we didn't actually talk about this one yet, but very generally, the virus can attach to specific host cells, right? We talked about spike proteins and specificity of viruses. Once the attachment happens, we're going to see in the next section, the genome will enter the cell and will direct and kind of hijack the host cell machinery to start making viral components instead of doing their normal thing. The viral proteins and nucleic acids are going to assemble, and then those are going to be released to infect other cells. For the next one, differentiate among bacteriophages, plant and animal viruses. Bacteriophages infect bacteria. They have a pretty complex structure often, with a head and a tail. For plant viruses, they infect plant cells, and they might cause mosaic patterns like we saw in tobacco mosaic virus. For animal viruses, these infect animal cells, and they can be DNA or RNA viruses. They can be enveloped or naked, and there are many that cause human disease. And finally, the characteristics used to identify viruses as obligate intracellular parasites. Remember, they cannot reproduce outside of a host cell. They have to use our host enzymes, our ribosomes, and our energy for replication, our meaning the host cell, whether it's human or animal or another host. And they show host and tissue specificity, right? Viruses can only infect certain species or even only certain cell types within that species. They're identified by the presence of virions, by their genome type, by whether they have a capsid or an envelope, and also by their replication method. And that takes us to the end of our intro to viruses. In the next section, we're going to talk about their life cycles.